Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Kay Tai will present Lifting the Veil on Neurons that Assign Good or Bad Associations to Our Environment. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $394 million to fund more than 5,700 grants to more than 4,700 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists. I'm delighted to introduce Dr. Kay Tai. Dr. Tai is an associate professor in the Department of Brain and Cognitive Sciences at the Picower Institute for Learning and Memory at Massachusetts Institute of Technology. She is a member of our scientific council and was a 2013 Foundation Young Investigator grantee and a 2016 Friedman Prize winner for exceptional basic research. Today's webinar will begin with Dr. Tai's presentation. This will be followed by a question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. Feel free to submit your questions throughout the presentation. Following the presentation, I will ask as many questions as time will allot. And now, I'm pleased to introduce Dr. Tai. Kay, the floor is yours.
Thank you. Thank you so much for having me here, Jeff. It's an honor. Okay.
Excellent.
All right. I'm just starting up my presentation. You guys can see okay?
Excellent. Yes, indeed, we do.
Okay, so it's my great pleasure to be speaking to you today. Some stimuli carry innate emotional valence. No learning or training is required to have emotional responses to them. Other stimuli, such as this image, are initially neutral unless they're paired with an emotionally significant outcome, such as the horrific events of 9/11. And to have an emotional association to this image, we have to be able to form that association. In my field, it's been very controversial about in respect to whether animals and humans experience emotion subjectively in the same way. Of course, it is impossible to say for certain whether the subjective experience of an animal is similar or different to that of a human being. I would also argue that it's impossible to say that the subjective emotional experience of any individual human being is like anyone else's. So even though what I'm really passionate about and interested in is understanding how emotions are processed, what I actually study are motivated behaviors, the best proxy that we have. So the big question that my lab is interested in is how do we assign motivational significance to sensory stimuli? Or in other words, how do we know if something is good or bad? We'll take the example of Usain Bolt. So perhaps if we have a sound like a gunshot, and that gunshot is paired with the experience of winning a bunch of gold medals, perhaps that gunshot evokes sensations of excitement or of joy. However, if that same auditory stimulus, a gunshot, bang, is paired with negative experiences in the case of a war veteran, perhaps this could lead to a negative valence association. And so it's possible that this same auditory stimulus would evoke very, very different emotional responses based on our prior experience. Either positive or negative valence could be assigned. It's also important to note that if this same auditory stimulus is presented many, many, many times without any consequence or any significance to us, then we will develop a rapid habituation, and it won't evoke any sort of arousal or emotional state at all. And so here, what I'm illustrating for you is just very simple. It's an adaptation from the two-dimensional theory of emotion, where on the y-axis we have intensity or arousal. You might think about this as the physiological response, your heart rate going up, you're sweating, blood pressure increases. And then on the x-axis here, we have valence or hedonic value, just an indication of how we are evaluating this arousal. I also want, not to confuse you, but to contrast this emotional theory with another one, the two-factor theory of emotion. And the idea here is that a stimulus comes in, and the first question that we ask ourselves, or we answer for ourselves, is it important? Is this something that is salient, arousing? And you might think of that as the absolute value. If it is not, then it's neutral. If it is, then the next question that we ask ourselves is this bad or good? Does this promote avoidance or approach? And so this is what I want to use to define valence. And this is what I want to focus on today. How does our brain process this information? How do we send this information down one path or another? So if you think about information coming to the brain and then our subsequent evaluation, we're going to lead to very different behavioral outputs. Avoidance and approach are, diametrically opposed. This is, of course, a simplification, but just to illustrate this concept, the resultant opposing motor outputs need to separate somewhere in the neural circuitry. And so where is that? And how does that happen? That's the question I'm interested in too. So why should you care about motivational valence or emotional valence? So if you think of the brain working in this way as theorized, you can think of many different psychiatric disease states as being related to perturbations of valence processing. For example, and again, this is a very simplistic view, but just to get started on the conceptual framework, if you are an individual with anxiety, perhaps you have shifted the balance where more stimuli will be processed and interpreted in a negative way and relative to a positive valence way. And if you are an addict, perhaps one interpretation of this behavior is that you have too much motivation to seek out potential rewards in your environment and not enough motivation to avoid negative consequences. And then perhaps in cases like depression, this motivational scale gets perturbed in yet another way. Okay, so where do we begin? How do we begin to understand the neural circuits of emotional valence processing? So one brain region that I'm going to focus on today is the amygdala. And we've known that the amygdala is important for emotional processing of environmental cues for over a century, from the work of Brown and Schaefer, then Kluver and Busey, and then Larry Weisskranz, specifically locating this to the amygdala. We know that amygdala lesions produce an impairment in recognizing environmental cues as either positive or negative. And so, for example, this monkey is playing with a snake, which normally it would not do. And in humans, there's the famous case study patient, SM, who has bilateral amygdala damage. And she was well known to have lost the fear to snakes and spiders, the ability to recognize emotion in other people's faces, the ability to recognize potential threats upon herself. However, more recently, it's been shown that she still had the autonomic responses and was able to show panic-like physiological changes upon suffocation. And so this just suggests that the amygdala is important for interpreting the world around us and these different environmental stimuli, and then linking that to a physiological response. It's not in itself necessary for producing that autonomic response. And in my lab, we actually work primarily on rodent models, rats and mice, primarily mice. And if we take a coronal section through a mouse brain and turn it around like this is what the mouse amygdala looks like. And there are many different subnuclei of the amygdala. There are 13 subnuclei, in fact, but here I'll just focus on the basolateral amygdala and the central amygdala. Okay, so the outline for today's talk will be where do circuits encoding positive and negative valence overlap and diverge? And how do these positive and negative valence encoding circuits interact with each other locally? What are their dynamics? And then, although I won't have much time to go over this, when do valence coding circuits engage in bottom-up versus top-down processes? How do emotions engage with cognition, for example? Okay, so first, just a tiny bit of background about the amygdala. This is sort of the amygdala 101. Very, very briefly, there are many, many groups that have contributed to this, but I'll just simply say that the basolateral amygdala has been likened to the cortex in that it's 90% glutamatergic projection neurons, and associative plasticity can be found here. And then the central amygdala is thought to be striatal-like because it's largely composed of GABAergic medium spiny neurons. So they're, of course, different than the cortex and striatum, but some have likened it to a primitive analog of the cortical striatal loop or circuit. So why do we begin in the amygdala? There's several pieces of information that I want to quickly review so that you can understand why we're starting here. So first, we know that neurons in the basolateral amygdala have been shown to encode positive and negative valence. And I think electrophysiological recordings have shown that there are partially non-overlapping populations of neurons that will respond to cues that predict positive or negative outcomes. Secondly, sensory information of all modalities reaches and converges within the basolateral amygdala. And so that's, if we're thinking about this in the context of Pavlovian conditioning, where we have a conditioned stimulus, an initially neutral stimulus that we pair with a motivationally significant outcome, all of that sensory information does converge in the basolateral amygdala. And third, we know that learning induces synaptic plasticity within the basolateral amygdala. So just a little bit about that. Here we go. Okay, so specifically, it's been proposed that synaptic plasticity occurs on inputs carrying information about the conditioned stimulus, something that might be an initially weak synaptic input that becomes potentiated with associative learning. And so I'm just going to talk very briefly about this so you can understand some of our results. So AMPA to NMD ratio, this just describes different populations of glutamatergic receptors. But the idea is following your garden variety long-term potentiation, or LTP, the current standing cellular mechanism for memory formation, one typical readout for this is that there's an increase in AMPA receptor number or function. And after LTP, what we typically see is that the current mediated through NMDA receptors maintain some sort of stability while AMPA receptors change. So with LTP, you'll see an increase in AMPA to NMD ratio, and with LTD, you should expect to see a decrease in AMPA to NMD ratio. This is not a perfect measure, but it is a reproducible and relatively reliable proxy for glutamatergic synaptic strength, although it does have its caveats. So several groups have previously shown that if you train an animal to learn that a sound, that a tone predicts a foot shock, and so animals will learn to exhibit fear-related behaviors or defensive behaviors in response to this shock predictive cue. This is called Pavlovian fear conditioning. This produces an increase in AMPA to NMD ratio in putative thalamo-amygdala synapses, right here. As a graduate student, my graduate work showed that reward conditioning also increases AMPA to NMD ratio in these same putative thalamo-amygdala synapses. And so this, raised a lot of questions at the time when we were making this discovery. How is it possible that the same mechanism underlies both fear and reward conditioning? Well, I suppose there are multiple possible explanations. One being maybe the amygdala doesn't encode valence at all. It just encodes salience. So regardless of whether it's good or bad, regardless of whether it's positive or negative, as long as it is important, you will observe such plasticity. Another possibility is that maybe the amygdala is the site of valence assignment, but that this occurs via distinct projections, distinct anatomical projections, where the message, there's a fork in the railroad tracks, and there's a switch operator, perhaps, and this is a metaphor, which switches the tracks to sending information down one path versus the other. So we also know, of course, there are many, many, many different projections from the basolateral amygdala to many different downstream targets, and even within the downstream structures I'll introduce to you today, the central amygdala and the nucleus accumbens, there are many different cell types, and that is important. But for simplicity's sake, I'll just review a subset of the literature. We know that the central medial nucleus is critical for the expression of fear. If you optogenetically stimulate central medial neurons, work from Andreas Luthi and David Anderson's labs have shown that this evokes freezing responses. And also that if you disconnect the input from BLA to the central medial nucleus, you abolish fear expression as measured by freezing. Of course, it's not that simple. There are many other types of behaviors that the central medial amygdala is important for, but I don't have time to go over that for the moment. So please refer to these other authors. We also know, and there's far too much literature to cite exhaustively here, but just a few seminal studies that I want to note are that the nucleus accumbens is important for reward-related processes, and specifically that optogenetically stimulating BLA terminals in the nucleus accumbens supports positive reinforcement, self-stimulation, and place preference. And so several years ago now, when Praneeth Namburi, a former graduate student, and Anna Baylor, a former postdoc in my lab, came to the lab, they were hungry and ready to take on an ambitious and challenging project. And the question that we had was, what is the circuit mechanism for assigning positive or negative valence? And so our hypothesis, very simple, is that BLA neuron projection target predicts learning-induced synaptic plasticity, namely that after we pair a cue with a foot shock, we would see an increase in synaptic strength of these synapses going on to BLA neurons that terminate in the central medial nucleus. And for the rest of the talk, I'm only going to be talking about basolateral amygdala neurons, and I might refer to them as CEM projectors or NAC projectors, but when I say that, it means BLA neurons that are projecting to these targets. Conversely, if we pair a tone with sucrose delivery, a reward, what we would expect to see is a relative synaptic strengthening of inputs onto these BLA to NAC synapses. Okay, so what we actually found, what Praneeth and Anna did, was that they first labeled these neurons in the basolateral amygdala, very simple, just retrograde labels of basolateral amygdala neurons depending on projection target. We then trained animals on either a fear conditioning task or a reward conditioning task. And then we performed the same experiments that everyone else performed, except for we recorded from neurons knowing their projection targets. And as you can see here, they are relatively intermingled. And what we found was that following fear conditioning, and this is what we expected to see, that there was a significant increase in amplitude ratio or synaptic strength on animals that underwent this paired conditioning as compared to animals that were exposed to the same number of tones and shocks but in an explicitly unpaired fashion, as well as to naive controls, which did not receive any of these stimulations, these manipulations. So this is what we expect. Okay, great. After fear conditioning, we see synaptic strengthening. That's not particularly surprising. What we didn't expect to see was this depression, a reduction in synaptic strength following reward conditioning in these neurons that project from the basolateral amygdala to the central medial nucleus. That was exciting. Now, I'm going to show you the data for the other projection. So pay attention. So now I'm showing you synapses onto BLA to accumbens neurons, and these neurons show the exact opposite. After fear conditioning, we see a reduction in AMPA to NMDA ratio. However, after reward learning, we again see this double dissociation, where there's now an increase in synaptic strength and input-immune ratio. So opposing changes are happening in both populations of neurons and synapses onto these different populations of neurons, and that they are the opposite of each other. So just to, and as a point to foreshadow, one thing I want to point out is that for reward conditioning, we food restrict the animals to motivate them and encourage them to be able to learn the task in one day. And so the only difference between these two naive groups is that the naive FR group, that stands for food restriction, these animals have been food restricted before consuming the sucrose, the same amount of sucrose as the other groups they're controlling for. And although this is not what we set out to study, we did note that there seemed to be a change just from food restriction alone, and that leads to some of our new projects. Okay, so just to summarize this part of the talk, these are old data at this point, but there are these opposite learning-induced changes in synaptic strength upon each of these projection-defined populations of the amygdala neurons. So, of course, the next question is there a causal relationship? Because animals that live in the laboratory, they live in relatively impoverished environments, and when we present them with something really salient like a big bowl of sucrose or foot shock, this could be so salient that it could cause a lot of plastic changes throughout the brain. And so the question that we wanted to test was, if it's true that the amygdala represents a hub of sorts to translate sensory information into motivational significance, then we should be able to bypass all this plasticity, just stimulate these output neurons and produce motivated behaviors. And so in collaboration with Ian Wickersham, we used a rabies virus that contained either channel rhodopsin 2, a light-activated cation permeable channel that depolarizes neurons and causes them to fire action potentials, or rabies virus containing just this fluorophore called Venus. And so we injected these viruses and their control viruses into either of these downstream targets and then illuminated in the basolateral amygdala. And we found that animals would perform intracranial self-stimulation when they had expressed channel rhodopsin and BLA neurons projecting to the nucleus accumbens, but not for those projecting to the central medial nucleus and not for controls. We then looked at what happened if we performed a real-time closed-loop place avoidance assay. And this task is also very simple and commonly used. And basically, when the animals on one side of the chamber, they receive photostimulation, and on the other side of the chamber, they don't. And the animal's freely able to explore either side of the chamber and come and go as he pleases. And in this case, when we photostimulate the central medial projectors, contingent upon being on one side of the chamber, animals show us that they want to avoid this side of the chamber, consistent with a place avoidance that might suggest aversion. So that is what we found here. So again, we see these opposite functions. Now, importantly, if this was an NMDA receptor-dependent plasticity mechanism, and our typical standard LTP mechanism is the following. Glutamate is released. It binds to glutamate receptors. Glutamate receptors, specifically AMP receptors, will open and allow some depolarization, for example, influx of sodium. And usually at baseline, at resting memory potentials, there's a magnesium blockade that sits within the NMDA receptor pore right here. And so we need this depolarization to happen such that the magnesium blockade can come out and calcium and other things can go in to initiate the cascade. So if this is indeed an NMDA receptor-dependent mechanism, then we would expect that hyperpolarizing the postsynaptic neuron would prevent learning because it would prevent the depolarization required to remove the magnesium blockade from the pore. So in the next experiment, in this experiment, we actually used a dual virus approach where we injected a retrogradely traveling viral vector carrying Cre recombinase into each of the downstream targets. And this particular manipulation was done bilaterally. And an anterogradely traveling viral vector that is Cre dependent carrying halorhodopsin. And then we train animals in either a fear or reward conditioning task where we photoinhibited only during the presentation of the unconditioned stimulus, during either the shock or during sucrose consumption. And what we found was a little bit of a surprise. So what we found with fear conditioning was somewhat expected. We saw an impairment in freezing, in fear conditioning when we photoinhibited the central amygdala projecting BLA neurons, and no effect on the other populations. Now, the big surprise was here. What we found was not what I expected. What I would have expected would have been that we would have seen an impairment of photoinhibiting accumbens projecting BLA neurons. That is not what we see. As you can see here, the accumbens NPHR group is lying right on top of our EYP control groups, which are controls for the tethering, the surgery, the illumination, etc., and the heating. And what we did see instead, which was very surprising to me, at least, was that the central amygdala projecting BLA neurons, when we silenced them, we actually saw an enhancement of reward conditioning. And so this was interesting. We saw this bidirectional effect of one population and no effect of the accumbens projecting population. And so at the time that we published this paper, we just sort of said, well, maybe this is just, a technical limitation. We didn't target the entire accumbens, and that might still be true. There's also diversity of different neurons downstream. Maybe we require finer granularity to observe these changes. But the thing I really want to stress to you is that this is something that kept me up at night, and it's actually now the basis of a new project that I'll touch upon briefly. Okay, so lots of foreshadowing. So again, just to summarize what I've told you so far, it's a lot of information. First, that there are opposite synaptic changes that map onto projection. An increase of synaptic strength following fear conditioning, and an increase of synaptic strength onto accumbens projectors following reward conditioning. We also know that if we just forget about all these inputs and all these synaptic changes upstream, and we just manipulate these populations of neurons, we can activate these projections and cause either approach or avoidance. And then finally, the weird thing, the thing that kept me up at night, was that inhibiting CEM projectors impaired fear conditioning but enhanced reward conditioning. There's this very asymmetric effect. And so we're not sure, this is something that we revisit. So on the surface, there's these projection-specific relationships with valence coding. It sounds simple. I'm trying to highlight some of the complications in the biology here. And that was a question that we asked ourselves. Is it really that simple? And, if there's any theme in biology, I think the general theme is that it's never that simple. And in fact, in the same year, we wrote a review. I had the privilege of writing a review with my former PhD advisor, Patricia Janik. And one of the things that we wanted to stress was that although genetically encodable tools for neural manipulation allow far greater targeting specificity than before, it is unlikely that all of the neurons that project from one region to another or that share genetic marker have identical functions. Thus, we may still be observing a majority vote for a given behavioral readout when manipulating any circuit component. And the thing I want to stress about this is oftentimes people conflate some concepts in neuroscience where if you stimulate a projection and you get a behavioral output, that it must mean that this is what all the neurons that you stimulated do. But that is equally logically flawed to saying that because we have one president, everybody in the United States agrees who it should be. And so the brain is similar. Many neurons have heterogeneous response profiles, but yet there's one behavioral output. And so that's why we compared this to a majority vote. Looking back, of course, we wrote this review in 2015. Looking back, hindsight's 2020. I kind of wish that I rewrote this article and instead of wrote about it being a majority vote, considered the possibility that not all votes are equal, more like an electoral college where some votes are more influential than others. And so I'll come back to this concept of the electoral vote in just a moment with some data that align with this notion. But first, I want to bring it back to a quote from Eve Marder. She says it so well, very simply. Optogenetic tools tell us what neurons can do, not what neurons do. So, okay, the next thing we wanted to do was to record from all of these neurons defined by projection. What is each projection-defined population of neurons encoding? And in this study, led by Anna Baylor, we wanted to explore this question. And so again, I still have not quite answered this first question. Where do circuits encoding positive and negative valence diverge? Is it really that simple? How heterogeneous are the coding properties of these neurons? And so first, in terms of calling something valence encoding or not, first I want to define this concept. There's a minimal criteria for valence encoding in single cells. And here I'm plotting the possible responses that you could have to a reward predictive conditioned stimulus. You could have an inhibition, the neuron could fire less, the neuron could not change, or the neuron could fire more. Same possibilities for a conditioned stimulus predicting punishment. And obviously this is also very simple, but I'm told this is the maximal complexity tolerated in a slide presentation. And so that's how I'll keep it for the moment. But first, the neurons must be task responsive. If the neurons don't respond to anything in the task, then they are not valence coding, provided that this is what your task is designed to do. Second, very importantly, you need differential responding. Neurons that respond similarly to both cues that predict rewards and cues that predict punishments are not coding for valence per se. I mean, perhaps there's a magnitude difference, but barring that, if the responses are similar, then you cannot call them valence encoding. They could be salience or arousal encoding. So it's important that you record under both of these conditions in the same cell to make that claim. And finally, we want them to be independent of stimulus features. There's some stimulus modality coding within the basolateral amygdala, meaning that some neurons receive only visual information or auditory information, et cetera, et cetera. So those are the things we want to be aware of. And so in this task, Anna Baylor, a former postdoc in my lab that now has her own lab in Bordeaux, and she's very talented, devised this task where a mouse was trained to lick for sucrose, following a sucrose predictive cue, and then another cue predicted quinine delivery, and the animal will receive that cue right here, and that the mouse will withhold licking in that case. And so we trained animals on this discrimination task, and then after the animal acquired the task, we recorded in the basolateral amygdala and performed photo identification. So before I get into photo identification, this is just a quick glance at all of the raw data. And I know you might be thinking, what am I supposed to make of this? Here, black is inhibited and yellow is excited, meaning more firing. And I'm just showing you all of the neurons that we recorded from. And what I expect you to take away from this figure is that there's a diversity of responses. And in fact, if we compare our data with the data of other studies in primates and in rats, performed by other groups and other people, specifically Dan Salzman's lab and Patricia Janik's lab, we see almost the same breakdown, the same proportions of neurons that do each things. So it's nice that we were able to reproduce these initial findings with high throughput recordings. And again, Anna recorded from over 1,600 neurons. But the point of doing this was not so that we would just reproduce the data, although that's always nice. We wanted to be able to identify specific neurons. And so using the same dual virus approach as we used before, we can express channel rhodopsin into projection-defined populations of neurons and use a technique that is often referred to as PhotoTagging, originally termed photostimulation-assisted identification of neuronal populations, developed by Susanna Lima and Tony Zader's lab. And I might refer to this as phototagging or photo identification. And the idea is simple. We've got our optic fiber, we've got our electrode. Neuron A fires, we record that. This is during the behavioral task. Neuron B fires, we record that action potential as well. Then after the recording session, only after all the behavioral, the task is done and the recording of the natural responses of the neurons is completed, we then shine light and identify which neurons respond. So we'll have neurons that are not responsive to light and neurons that are photoresponsive to light. And this is just a peri-event raster histogram demonstrating light responsive and non-light responsive neurons. So again, green photoresponsive neurons here would be a good example of a neuron that's phototagged. Of course, it's not that simple. There's this major caveat of recurrent excitation. I'll try to briefly introduce that to you here. So the idea is, okay, we've got our channel rhodopsin expressing neurons. We're very confident about expressing channel rhodopsin in them using these viral vector tools that have been used by hundreds of groups. And it's easy to tell this CTR2 expressing neuron that will respond to light from this non-expressing neighboring neuron that doesn't receive any input and does not respond to light. That's easy. Much more difficult is how do we differentiate this population of neurons from non-expressing neighbors that do receive indirect input that may not themselves project to the target that we are examining. And so, in short, what we can do is record from these neurons, patch from them, and know with confidence whether they whether they express channel rhodopsin or not, and then systematically survey the latencies, the amount of time that it takes between the light pulse onset using different light powers and the action potential that we expect to see. And so, in this particular study, we were very fortunate. The distribution of photoresponses for non-expressing neighbors was much higher than that of the photoresponse latency for neurons that were expressing channel rhodopsin. So in this case, it was black and white and very easy. However, it's possible that you'll have cases like this where there's some overlap. This is not the case in this particular study, but something that we should all, take caution to be aware of. And in that case, you could draw the threshold here, but then you're having a lot of false negatives. And then there are cases where the distributions overlap to such a high degree that phototagging may not be the best strategy for this technique. And all of these different things depend on the details of the experiment, the incubation time of the viral vectors, expression levels, light levels, neuronal type, the biology in each unique preparation. And so this is just something that I think everyone should be testing in their own experiments. Okay, so I won't go through all the details of this because this is published. I just want to summarize by saying that what we found was consistent with our initial predictions, that BLA2 nucleus accumbens predictors predominantly encode positive valence, and BLA neurons that predict the central amygdala predominantly encode negative valence. And this is true if we compare on a single neuron by neuron basis their difference of responding to cues that predict sucrose or quinine. And also if we look at overall qualitative examinations of whether they were excited or inhibited by these different cues. And so while the overall population has a bias towards positive or negative valence coding, as you can see here, this bias is not heterogeneous. This is not homogeneous. The neurons are very heterogeneous, and there are certainly many exceptions within each population. Okay, so I hope I've convinced you that the BLA is a site of divergence for positive and negative valence, and it's a very important site. And now that I've introduced a lot of these concepts to you, we can ask how these positive and negative circuits interact. Why are they all intermingled together? Why would the brain be designed this way? So one clue about local interactions. So one idea of why these different projection-defined populations of neurons are so intermingled with each other is that they could interact. And we have empirical evidence that they interact. So in these same recordings, here I'm showing you population data from the central amygdala projecting population of BLA neurons. In the top row in red are the phototagged neurons, ones that we have deemed to be CTR2 responsive based on our very stringent criterion. And then in black are neurons that were also excited in response to light, but did not meet our photo response latency thresholds or other criteria. And then finally, this other population of neurons that was photoinhibited, meaning when we activated these neurons that projected from the BLA to the central medial nucleus, these other neurons in the BLA were inhibited and silenced. And I just want to point out that the number of neurons that are photoinhibited is greater than the number of neurons that are activated, despite that manipulation that we're doing is an excitation and activation of these neurons. And so this led us to back to this idea. So if you recall, I was just talking about this notion of an electoral college where not all votes are weighted equally. And here is the biological data So this is, if we did the same thing for each projection, the colored, this is normalized for the number of neurons that are identified as CH2 expressing. In black are the neurons that are photoexcited, so network excited neurons, and in gray are the neurons that are inhibited by the neighboring, by their neighbors. So indirectly inhibited. And what I want to point out is that these belated central amygdala neurons have a very strong capability to suppress their neighbors and to influence the activity of other BLA neurons, much more so, significantly more so than these other projection-defined populations of neurons. Again, supporting this notion that not all neurons have the same amount of influence over other neural activity, over behavioral output. Okay. So we next wanted to examine the arrangement, the architecture of these neurons. Does form give rise to function in some way? So what I'm showing you here are these neurons that we've labeled and that are intermingled. And I'm going to show you a video where we've cleared the brain. And here, green are nucleus accumbens neurons. Blue are these central amygdala projecting BLA neurons. And you can see that these neurons are somewhat intermingled with each other. There are gradients. There is some topography, but there isn't any sort of segregation or divide. And so this brings us again back to this question of how do these neurons actually interact? And so we thought the possible connectivity would be mutual inhibition. It makes sense that reward encoding and fear encoding neurons might inhibit each other to aid action selection. That was actually what I would have initially put my money on. At the time I wrote this review, at the time we were drawing this out, I would personally not have predicted that unidirectional inhibition would have been the mechanism. It was just technically another possibility, so we included it. And then we also know that the amygdala is very epileptogenic, so there is some recurrent excitation that happens. So we thought there could also be a neutral or excitatory relationship. And just to give you some context about why you should care about this next bit of data, and this is my last story that I want to share with you today, is this idea that homeostatic need state dramatically influences emotion, which in turn influences many other processes, including perception, action selection, decision making. And so the example here is that these data are taken from judges in Israel that were making decisions. This is over 1,000 judges that were making decisions about parole. So they're hearing different parole cases throughout the course of the day. And these are people that have trained their whole life to make unbiased, fair judgments that are obviously very important to other people's lives. And the favorable parole decisions were at 65%-ish at the beginning of the day, plummeting rapidly to single digits right before the first meal break. Then judges come back from the meal break, back up to roughly 65%, and then again dropping rapidly. So it seems that the time at which your case is heard is more important than perhaps your case if other things are distributed equally. So it's kind of a wake-up call that homeostatic need does indeed influence emotion in a very important and powerful way that we should be aware of. The work I'm going to tell you about, this is my last story, has been led by Gwendolyn Calhoun, Amy Sutton, and Jarong Chang. And here I'm just trying to simplify this diagram. Again, these are BLA neurons that project either to the nucleus accumbens or the central medial nucleus. I'm just going to abbreviate them. Again, they're predominantly driving either approach or avoidance, and I'm going to abbreviate them with just an N and a C for simplicity's sake. Now, in the naive condition, what we have here is on the rows, we are stimulating these neurons, and on the columns, we are recording from these neurons. And what we found is that when we stimulate from central amygdala projecting BLA neurons, we see a suppression in firing in accumbens projecting BLA neurons. That's what we expected. Sort of a facilitation within population, mixed bag within population here. And then the very confusing piece of data for me was when we stimulated accumbens projecting BLA neurons and recorded from central amygdala projecting BLA neurons, that we would see a facilitation. And this was very confusing. Why would when we stimulate these largely reward-related population of neurons, why would we see a facilitation of largely avoidance or fear-related populations of neurons? This was very confusing. And we did this experiment many times with many different parameters, and it is a very robust result. And so we were confused about this asymmetric unidirectional relationship. Why would this make sense? Previously, I showed you a symmetric change, but I didn't mention that there's food restriction. So why would this make sense? How does this fit with everything else we know? Again, there's some asymmetry in our previous data that I pointed out to you here and here. But what I really want to say in terms of why the brain might have evolved this way is that if animals are engaging in social behavior, in feeding behavior, any other type of reward-seeking behavior, almost invariably they are more vulnerable to predatory threats, especially in the case of mice that normally live in the safety of their burrow. If they go out to forage for food or water or mates, reward seeking is inherently risky. So perhaps priming escape is a good insurance policy. And I also want to mention, again, back to this issue of homeostatic need, and I do promise this is my last bit, that animals will normally behave in certain ways, but a lioness, for example, would rarely attack an elephant of this size in broad daylight without the rest of her pride. But in conditions of near starvation, she might do so. Baboons would rarely forage in water for risk of being attacked by caiman, but in resource-scarce conditions, they will do so. And so we change our behavior when we are in different homeostatic need states. So if we food deprive the animals for not even one day, just 20 hours, so not even one full day of food deprivation, and repeat this very same experiment, we get a very strikingly different result. Before, we got a suppression in this particular panel. When we stimulate these central amygdala neurons and record from Cummins projectors, we previously saw a suppression. Now we see a facilitation, and a facilitation exists all around. So just to summarize this part of the talk, and the final thing is that this is the summary of our ex vivo ePhys data. I do want to point out, how do individual cells change across homeostatic states? And so Jiarong Chang in my lab has pioneered this longitudinal imaging of projection-defined populations of neurons in the basolateral amygdala across different need states. And what she has found is that if we look at BLA to accumbens neurons, that in the SATA-deprived case, here on day one, they're relatively low level of activity. In the food-deprived condition, these same neurons have more calcium transients, which are proxies for neuronal activity, and this returns back to baseline once we refeed the animals on day. We actually give them an extra day, day 4. And so here, that's what those data look like. You can see this robust increase in activity with food deprivation. If we do the same exact experiment, but now look at BLA neurons that protect the central medial nucleus, we see the exact opposite. And I want to put these data up against each other because if you look at the baseline data, they're almost identical. If you combine these neurons and did not look at the projection target of them, you might not see any change at all. The changes might just wash out. But what you can see here is that there are these opposite changes that are carried by many of the neurons within the population that change with food deprivation. And so finally, to conclude and put this all together, the relationship between these positive and negative valence coding neurons is state dependent. And we see that the activity of BLA to accumbens neurons increases and activity of BLA to CEM decreases after food deprivation. So compared to what is theorized on our two-dimensional plot here, with food deprivation, in our naive condition, it might not actually be accurate. It might not be equal, that positive and negative valence coding circuits are symmetrical in their coding, in their strength. But when you food deprive animals, this shifts the balance, shifts the fulcrum, and might alter the relationship between these neurons. And this is one mechanism for how homeostatic need states can influence valence coding, the way that we evaluate the world around us, and the way that we behave. So with that, I will just close and summarize and say, again, I hope I've convinced you that the amygdala is very important. Whether you're a basic scientist and interested in understanding the fundamental building blocks of learning and memory, or you're a clinician and interested in the translational properties, the fact that the amygdala circuits are so well conserved across evolution, from a translational perspective, this increases the likelihood that insights that we observe in mouse models might also be relevant for humans. And So with that, I've tried to acknowledge everyone along the way. I want to acknowledge all the other members of my laboratory, our collaborators, and our funding sources, and you for your attention. And I'm happy to take any questions that you might have.
Well, Kay, thank you for an excellent presentation. The work that you're doing is extremely important and complicated, but you did a good job putting it in as lay-friendly terms as possible. So thank you for doing that. One question relates to what you started to speak about at the end, which is how insights in the mouse may be relevant to humans. And I'm curious about your perspective in terms of how some of these findings might relate to conditions that humans have, such as post-traumatic stress or some of the other conditions that are so important that we treat.
Great question. And so I didn't have time to go into all of the details about how we decided where to look and what to examine. But in one of the studies that I actually didn't discuss today, looking at anxiety-related behavior, there was a human functional resting state imaging study performed by Amit Etkin, where they found that there was an abnormality in patients diagnosed with generalized anxiety disorder in the functional connectivity between the basolateral amygdala and the central medial nucleus. And so that has been one of our inspirations for further targeting this projection in the context of aversive learning as well as innate anxiety. And so that is just one example of how well conserved these circuits might be. And I think really what I would like to push for is to take more of a circuit-based therapeutic approach, using what we know about circuitry and designing potential therapies using based on those fundamental principles.
So taking it to the next step, would the type of approach that you're speaking about allow for testing, let's say, potential medications in mice in these circuits as being a more direct relevance to humans, given how you describe the relationship between these circuits in mice and in humans?
So I think I see really two main possibilities. One possibility would be, now that we know that these different projection-defined populations of neurons, and maybe we can get even into a finer level of granularity, but using this general type of approach, we can then identify neurons based on function and then perform, for example, transcriptome profiling or sequencing to look for unique surface receptors that might represent novel targets or new targets for small molecules for example, pills that could be taken that are mechanistically novel. Another approach, maybe, that would require a bit of luck. Probably not, it's certainly still feasible. Another possibility is that we develop more non-invasive therapies. And this is actually something that I'm working on in collaboration with a number of other investigators, including Amit Etkin, to be able to develop non-invasive ways to target certain circuit nodes and induce plasticity. Since we know that plasticity happens naturally, and we know that we can change the transmission between certain synapses using various tools, what if we were to change the strength of certain synapses in a long-lasting manner? Could we then have a long-lasting therapeutic effect? Right now, we have to take a drug, every day, and it's sort of a Band-Aid, right? It's not curing the disease state; it's treating the disease state. If we identified that the disease state was indeed a perturbation in, for example, synaptic strength, perhaps we could actually cut to the jugular and really solve that problem by treating the core feature that defines the disease, and then really develop a cure instead of just a therapy. So that's the hope.
Which is obviously our hope is, you just said it in a nutshell, to develop cures for these conditions and even methods of prevention before they become full-blown. Is there, what would be the type of mechanism that could be used to have this result to really cure these issues?
So at this point, at this specific moment in time, at this moment, there is not yet a way to non-invasively target specific circuits deep within the brain. That is something that we are, might be just a few steps away. We are able to target surface areas of the brain using transcranial magnetic stimulation, focal ultrasound, red wavelength light shifted tools might become a possibility if viral vectors can be proven safe, which is perhaps maybe a few more steps away. But I think these things in combination with some cognitive behavioral therapy could really enhance the plasticity that occurs and then help reset the brain into a circuit state that is healthier.
So, for example, you bring up cognitive behavioral therapy. There's some research that shows that treatment with cognitive behavioral therapy has that ongoing effect, unlike taking a medicine, which if you stop taking medicine, it may no longer have the effect the cognitive behavioral therapy does. Perhaps the mechanism of action is the plasticity that results and changes that result from the therapy are permanent and therefore continue even after the therapy is ended.
Exactly. I think that's the idea. If you're inducing new plasticity, maybe you might call it learning. There's a lot of things that can induce plasticity, and maybe we can also nudge plasticity along in a certain direction in conjunction or in combination with cognitive behavioral therapy. That is one idea that I can see working out. If you think about right now for treatment-resistant patients with major depressive disorder, we still in 2018, we still give electroconvulsive shock therapy. And this has obviously no specificity. We're just, running current through the brain. And, the speculation about why this works is that you're inducing plasticity all over the brain. And here it's non-specific, so there are a lot of undesirable side effects. But you're inducing plasticity through the brain and then increasing, maybe there's some hope that the brain will then settle back into a stable state that it can maintain. And that's why ECT still works for some people. Obviously, again, it's non-specific. We're not aiming it at the specific circuits that require it. We're aiming it everywhere. And I think that's a huge problem. I think same thing with GBS. Sometimes that can be tremendously effective, and other times less so, because we are not specifically targeting the circuits that require it. And it's more, I think the greater the specificity and the more that we know, the greater the likelihood that we'll have effective treatments or even cures that don't have any undesirable side effects.
So you'll tell me if this sentence is correct, because I find it to be potentially very exciting, which is that the work that you and others are doing with mice can help lead us to pinpoint treatments in humans down the road, because by understanding these circuits, we then know where to go after humans to give that more pinpoint kind of treatment.
Exactly. I think there are a lot of parts of the human brain that have evolved that are totally different from the mouse brain, but a lot of those are higher cognitive functions. Our cortex is really what has evolved. These primitive amygdala circuits are largely conserved, much more so conserved. And I think that within the limbic system, where which I believe to be the seat of most psychiatric disease states, how do we find that needle in the haystack? And I think, using mouse models allows us a higher throughput way in circuits that are relatively conserved to try to discover what that key node or key circuit component is that we want to target. And then we can begin bringing it back to informing therapies for humans. But I think this basic science discovery is really, really important for us to get out of the rut that we've been in for mental health treatment development, which is essentially trial and error. And, I think we've been stagnating in that for several decades. And I'm excited about moving into a new era of potential therapy where we're informed by basic science. So that is my vision. That is my hope.
Well, I think that vision and hope for you offers a lot of hope for many, many people. And I want to thank you for, first of all, for joining us today, and more importantly, for all the work that you have done and continue to do. I think one of the things that's striking in your presentation is when you point out some of the people who have worked in your lab and have now moved on to their own lab and are continuing, how so much more research blossoms just from one researcher. And you mentioned mentors that you've had that now have brought about all the work that you're doing. And it just, I think, gives tremendous hope to people about the future for all of us. So I want to say thank you very, very much for all that you do.
Thank you. Thank you.
I also want to thank everybody for joining us today. All of the research we fund is made possible through private donations. So please consider making a contribution by visiting our website, bbrfoundation.org, or calling us at 800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it with a family member or friend, please visit the events and webinar page on our website. I hope you'll join us again next month when Dr. Dorothy Sitt, Associate Professor of Psychiatry and Behavioral Sciences at Northwestern University's Feinberg School of Medicine, will present Bright Light Therapy for Mood Disorders, including bipolar depression. This webinar will take place on Tuesday, September 18th at 2 p.m. Eastern Time. Thank you again for joining us, and enjoy the rest of your day. Take care.